So the slides will be up. Welcome, ladies and gentlemen, to the final presentation at this 22nd ISMB. The presentation from Russ Altman, who is an MD and a PhD. He's a professor for bioengineering and genetics and medicine and computational sciences in Stanford University. Russ is one of the people who brought us ISMB. It started with Russ. It started in Stanford in '94. That was one meeting that Russ chaired. He also was ISCB president from 2000 to 2001. He's one of the first ISCB fellows. He's a member of the Institute for Medicine and is a chair of the FDA Science Board. He has been a president of other associations. He has published. He has done so many things that I'm not going to take his time and give the floor instead to him. And Russ, he gave me a clue what I should also write. He says, I'm happy to be here. Wonderful. We're happy to have you, Russ.
Well, thank you very much, Burkhard, and it's great to see you all, and I'm stunned that you all are here and grateful. ISMB 1994 at Stanford had 220 attendees, so they would all fit pretty much over here. And so that was a good time to organize this meeting. It didn't take that much work, and it was a huge amount of fun. It was the second ISMB. I'm talking today about informatics for understanding drug response at all scales. I'll tell you a little bit about why I care about this, and then I'm going to tell 4 stories, 4.5 stories. The reason I do 4 stories is this is our discipline has grown into a very diverse group, and any single story might not be of interest to a large subset of you. So at the risk of appearing superficial, because you can't get into as much depth as you'd like to, I'm going to tell 4 short stories which will hopefully whet your appetite and get you excited about the potential for informatics, not only for understanding drug response, but then for doing things like discovering new drugs and new ways to use old drugs. So today, this is a little bit of an outline. The focus of our work at Stanford is on the drug action at molecular, cellular, and organismal levels for many purposes, including pharmacogenetics, mechanism of drugs, repurposing, interactions, side effects. One of the great things about informatics is that we are not wedded to a single scale. And I would encourage you to think about this in your work. We can work at the same time at the molecular level, angstroms, and at population health level with information about drug response of patients. That's about 9 or 10 orders of magnitude, depending on how you do math. And that is a special capability of our field, not necessarily easily available to our experimental colleagues who are great and who we love, but are typically specialists at 1 to 2 orders of magnitude of measurement. So that's the real message of my talk today, is that I'm going to tell you a story at the population level, mining databases about patients, the literature, which is some weird length scale that I can't really characterize, cells, and then molecules. And there's lots of things in between involving tissues and organs, but these will be the 4 stories that I tell, and hopefully will transmit to you my excitement about the possibilities for networking data at multiple levels to do amazing things. So a lot of this comes from the fact that my group for the last 14 years has been building the pharmacogenomics knowledge base, or PharmGKB. Please visit it because we need to count our hits. And so as long as you're not from a Stanford IP address, it counts. PharmGKB is charged with collecting all information about human genetic variation and how it impacts drug response. We've been doing it for 14 or 15 years. We're proud of it. Please visit it. This is all I'm going to say about it pretty much, except that it gives us this amazing platform to justify spin-off research projects to either understand phenotypes associated with drug use or the molecular mechanisms of drug response. So it's been nothing but a great experience building this database as a service activity, but then spinning off the types of projects that I'll be talking about for fun and research and discovery. This is one slide from one picture from the site, which is just to say that PharmGKB's most popular feature are these lovingly curated pathways that our curators build for both the pharmacodynamics, how a drug works, as well as the pharmacokinetics for how a drug is metabolized. And we put a lot of work into these pathways. The supporting data is all downloadable. The graphic itself is downloadable, and we let you use it in your, if you'd like to, in your publications. A couple of years ago, we used PharmGKB to annotate a complete human genome. It was the genome of a colleague of mine, Steve Quake, in the Department of Bioengineering, and we showed that in the case of pharmacogenomics, there are hundreds of drugs for which human genetic variation may actually influence doctors' decisions about how to use those drugs. Previous to that, there was a sense that, oh, isn't it one or two drugs and then not too much? And in this paper and in subsequent work, we've shown that no, there are tens to hundreds of drugs per person where the genetics might have a major influence on the response. In fact, to deliver on that promise, we've been involved in a consortium called the Clinical Pharmacogenetics Implementation Consortium, or CPIC, that actually publishes prescribing guidelines for physicians who want to be early adopters of pharmacogenomics. And in these papers, and we've had about 15 of them, and there's a long list of other papers coming out, we tell a practicing physician, here's the genetic variants that you might observe, here's how to transform those into a predicted phenotype, and then here's the dose of the drug or the adjustment to the dose that you should use if you want to use pharmacogenomics. So this is for us very exciting because we will certainly stop here. There are others who need to take this work forward, but the culmination of PharmGKB work in many ways is the creation of a clinical guideline advising how to use genetic information for drug response. So here's the first of the 4 stories, and the first is about mining population data sets for drug effects. Some of you may know that the FDA releases every quarter all reported adverse events from the drug companies, from providers, pharmacists, even from patients sometimes. And these provide noisy but valuable data about adverse events of drugs. We think they're an opportunity to find new effects of drugs, and I want to tell you about some work published now a couple of years ago, and so I'll talk about it quickly, where we asked, could we use this population-based data to generate new effects for old drugs or new interactions between drugs? Now, what an FDA report looks like is it gives you the sex of the patient, the age of the patient, all the medications they were taken, all the diseases that they have been diagnosed with, and when I say all, it's the best effort of the person reporting, so of course that's a noise right there. All the side effects that they experienced, and some measure of outcome in 5 big bins, like death, hospitalization, lost work. Many people consider this database to be a complete mess and not useful for discovery, but Nick Tatnetti, who was the first author of this paper and who's now at Columbia, didn't agree. The big problem that I'm sure you all would immediately guess is the issue of confounding effects. And just to give you 2 of the very most common confounders in these kinds of observational databases, if you know that drug X causes a side effect A, and if you know that drug Y is used along with drug X frequently, and this happens because of practice patterns of physicians all the time, then drug Y will appear to cause side effect X with a straightforward, not very thoughtful application of correlation. And this database is filled with that potential for synthetic association of a drug with a side effect that it doesn't cause, but which another drug causes. Even tougher is if a disease B causes side effect C, and drug X is used to treat disease B, you might infer that that drug causes the side effect. Again, this is really basic stuff, but this kills you when you try to do an analysis of these databases. In fact, think about diabetes causing hyperglycemia, high glucose levels in the blood. A straightforward application of discovery statistics to this database will tell you that every single diabetes drug is associated with high glucose. And it's not, of course, because the drugs cause high glucose. In fact, they're designed to lower the glucose, but they have this strong synthetic association. No surprises there. It's the transitivity of correlation. We came up with a way, and I'm summarizing it incredibly simply here. You'd have to read the paper, but it's kind of an obvious idea from the case control tradition, where you need to pick control drugs that have a similar expected correlation structure. And the surprise here is not that we did this, but how well it worked, and I'll show you some of the data. So when we compare a drug looking for its side effects, that we use not all other drugs in the database as the control, but other drugs that are used for similar diseases or that are used with other similar sets of drugs co-prescribed. And with a huge database like the FDA is releasing, by the way, I should say there are millions of records in these databases, we thought that you might be able to cancel out some of these associations. So what you have here on the left is, on the x-axis, it's the empirical level of correlation observed for a drug that is co-prescribed with a causative drug. And as you can see, that as the drug as the second drug is more and more correlated in its use with the first, the chances of seeing one of these synthetic association goes up. When you use these control groups, it flattens out, and that's what's shown on the right. No detectable bias. Similarly, if you look at shared indication, this is the diabetes example. If you look at drugs that are used for, that are correlated with use for a specific indication, the chances of a synthetic association skyrocket as the correlation, as the strength of the correlation goes up, and then that can also be eliminated. We found, to our surprise, that this also eliminates other things. There are correlations from age. There are drugs that old people use, and if you're not careful, you will assume that all the diseases of the elderly are side effects of these drugs. You can cancel out the age effects simply by using the co-prescribed drugs and the co-indications, not using any information about age specifically, but you can see a flat graph there on the right. And here are some actual examples. We have drugs that are mostly used in the young, or in the old. The dark filled-in boxes show the correlation that you might observe with a simplistic analysis of the data. So all those green dots on the left are used in young people, and most of them, when you correct, become those white circles at the line of indifference, if you will. There's no observed signal. The ones on the right are used in old people. There are all these signals that look artifactual, and when you correct, again, most of those circles come back to the 0, which means no significant difference. Now, there are some that are interesting. Let's look over here at retinoic acid, which has a signal, and when you correct for the age bias, the signal remains. And so this was great, because then we say this is worth reporting, worth following up. And so we write papers now about signals that persist even after these corrections. Fortunately, most of these corrections make most signals go away. And finally, there's an implicit correlation with the sex of the patients. So there are obviously certain drugs that are used in men more than women, and therefore those drugs are incorrectly often associated with manly side effects or womanly side effects. And we can get rid of those, and here's again an example of drugs that seem to have these signals in the dark green boxes, but those signals in many cases go away after correction, but in some interesting cases don't go away, and then you're off to the races potentially with discovery. So to make a long story short, we released 2 databases doing this and in some more sophisticated things, but that was the basic idea. Offsides is a database of off-label side effects of drugs based on analysis of this data, and we reported 438,000 new side effects that we saw evidence for in this database. A subset of which, I should say, we validated in electronic medical records to show that this was not hocus pocus, but many of them, most of them validated in electronic medical record, the small subset that we checked. We also released a database called Two Sides, which is a database of drug-drug interactions inferred from the FDA database with 868,000 drug-drug interactions. Now, and these are available at PharmGKB. My favorite part of this work was when a reporter called me up and said, Dr. Altman, you've reported 1.2 million new side effects. What do you expect the FDA to do with that? And I was embarrassed because I fumbled and I said, well, they should prioritize based on what's the most important side effects. But I didn't have a good solution, and I felt like I should be part of the solution. Oh, and I'm going to skip over this, but this is a spectacular picture showing a lot of side effects and a lot of drug interactions. It's very exciting, but skippable, and mostly colorful. Okay, so we said, oh, let's just get a list of side effects and how bad they are, and then we can rank order all these predictions by the really bad side effects that really should be paid attention to by the FDA. Well, guess what? There is no rating scale of side effects. There is a standard vocabulary, about 3,000 terms: cough, completed suicide, rash, skin falling off, and 3,000 others. But no one has ever said, these are bad, these are good. And so I couldn't quickly triage my list, and so this led to a project that Asaf Gottlieb in the lab is doing, where he used the crowd to rank side effect severity using Amazon's Mechanical Turk. This is a place where you pay people who evidently have a lot of free time one small amount of money to do a simple task. So let me tell you what we did. We generated, so we do have about 3,000 side effects that we wanted to rank, and we generated small lists of pairs of side effects, and we asked the workers, these are not health professionals, these are people who have time to make money on the internet. I guess they're not good at playing poker. And we asked them which side effect is worse. And we give some links, a Google search or something, so if they've never heard of the word, they can get a quick definition. And because we were worried about quality, we also have spiked in 2 side effects that we have decided which one is worse already. And what we do is we put in a few of those into every batch. So if they're giving random answers, we can tell because they'll get those wrong, and they're supposed to be easy and obvious. So we send these out. We get back a bunch of pairwise comparisons. A is worse than B. And then in an algorithm, so first I'll tell you that we had a couple of thousand workers. Most workers do a few comparisons, so we had like 800 workers here that did 10 or less. Some workers really like this task, and they did hundreds of comparisons. Most workers were doing a good job. It turns out that the Mechanical Turk people keep track of the quality of their workers, and they block out bad workers. So most people passed our quality control, to our surprise. And this shows the quality as a function of how much work they did. And the people who did a lot of work were actually quite high quality. In a method that I won't describe, because this is actually a very interesting area, how do you get a rank order from a set of pairwise comparisons? Good algorithm, not going to talk about it. We did a linear programming-based method to come up, and it's one of many, and we're going to release this data as soon as the paper comes out so that you can all beat this. But it allowed us to get a ranking using basically a linear programming approach. So what you see in the top left is the first batch versus the second batch correlation, about 0.75 correlation. So I said, well, that's good, but how do we know it's really robust? Well, we just did another one. And each of these experiments cost me about $1,000 and gets done in hours. This is really the amazing thing, it's not so much a nickel per comparison, it's that in 3 hours we have all the data that we need. So we did 3 total runs just to make sure, and you can see 1 versus 2, 2 versus 3, and 1 versus 3 in these 3 plots, and they're pretty much the same. And they all have the same correlation coefficient. And most satisfyingly, if you look at the side effects that were judged as most severe, there's very low variance over the 3 runs in what their rank is. And the ones that are very mild. The ones where there's confusion, there should be confusion because they're all kind of average severity, and it's hard, especially for a regular old person who's on the internet, to tell the difference. But we have error bounds, and we feel pretty good about this going forward now to triaging our predictions, because we have some sense that for the really bad ones, which is what we need for triage, that we have pretty good data. So just for your edification, here are some classes of drugs. I apologize for the small font. Classes of drugs listed from those that have the most severe side effects down. So immunosuppressants, you won't be surprised, is the #1 dangerous drug class. Anti-inflammatories and anti-rheumatologic, Parkinson drugs, anti-epileptic, and cancer drugs, all kind of pretty bad in their side effects. What we're plotting here is the number of severe ADRs in the package label for these drugs. And then, contrast media, diuretics, lipid-modifying drugs, quite safe. Not a lot of bad side effects, although some. And then we've correlated them, but I'm going to skip that. This whole thing costs not including Asaf's very valuable time as a postdoc, this costs $6,000. 2,600 Amazon Turk workers, 3,000 adverse events. Those workers made 126,000 comparisons, a nickel per comparison, and so it costs $6,000. So I am a fan of this for certain types of activities. That's my first 1.5 stories, because it had both the side effects as well as the triaging. Second story is about literature mining. So we all know that PubMed probably has more like 25 million abstracts now. From my perspective, it's important because it tells me about the relationships between genes and drugs, genes and small molecules. And there's been remarkable advances in natural language processing that allow us to not just look at co-occurrence, not that the gene and the drug are in the same sentence, but actually dissect what is the relationship between the gene and the drug to get a semantically much richer understanding of gene-drug relationships. And so we asked, can we characterize the full universe of gene-drug interaction modes? And I think the answer is yes. This is work done by Beth Percha, a graduate student in my lab, and the work is under review. So the Stanford parser has been created by my colleague Chris Manning in computer science at Stanford. It's a spectacular tool. It's freely available to all of you, so I recommend if you're interested in this that you use it. And when you give it this sentence, CYP3A4 mRNA expression was significantly increased by rifampicin exposure in human hepatocytes. It not only does a parse, but it creates a dependency graph. What you're looking at is a dependency graph that shows usually the verb at the top with the results of the parse showing kind of the noun phrase, the subject of the sentence, the object of the sentence. In this case, it has recognized that this is a passive voice sentence, which, by the way, you should never write. mRNA expression was increased. So it knows that this is the noun, subject, passive. There's an auxiliary verb. Here's the verb. Here's the agent. All of these, of course, have meaning. And it gives us a map of the sentence. Most importantly, the gene that I care about, CYP3A4, is here. And the drug that I care about, rifampicin, is here. And there's a path that connects them, which is shown on the next slide. I've taken away all the other decoration, which, by the way, is semantically useful and is a subject for future work. I'm not saying it's not useful. But there is a vector that connects 3A4 to rifampicin, which is expressed as a vector below, and which you can see graphically that there's a verb, there's a couple of other nouns, and then there's the gene and the drug. And we call that a dependency path. And it turns out that there are really not an infinite number of dependency paths or ways to express the relationship between a gene and a drug. There's a relatively finite, certainly there are modal ones that are the most common, and then there's a long tail of uncommon ones. For example, the most common dependency path we see in PubMed, this is now looking at every abstract for which we have text, about 270,000 sentences that have a gene and a drug in them. The most common relationship is to say drug, an inhibitor of gene. And that's APOS, the word inhibitor, and AMOD in that dependency path that I showed you. The second most common is APOS inhibitor preposition. So dicumarol, a potent inhibitor of NQ01. So, and then this goes on forever, down to a bunch of singletons. But for many of them, they are repeated several times, hundreds, thousands of times, in the PubMed database. So we've done this. We've parsed every sentence in PubMed that includes a gene and a drug. I won't go over all of these. But you can build this incredible matrix, which is a matrix of gene-drug pairs. So the rows are genes and drugs that appear in a sentence together. And the columns are the dependency graphs that connect them. You can either do the exact dependency graph or you can smooth it by removing and simplifying. We've done both. So there are about 50,000 gene-drug pairs in PubMed sentences. There's about 197,000 dependency paths. And what you see here is a bi-clustering of the genes-drug pairs and the dependency paths. So you can see one nice big bi-cluster here, a bunch of little biclusters everywhere. What does this mean? It means that the gene-drug relationship for the genes and drugs that are in these rows are characterized by very similar dependency paths, or to make it a little bit more colloquial, there are very similar sentences written about the relationship between those genes and drugs across all the literature. So we talk about those genes and drugs in the same way. And you can see there are hundreds of clusters here. Actually, I'll tell you how many we think there really are. So we did, and the biclustering algorithm is spectacular, and I'm not going to talk about it. We're very excited about that too. But one of the things that we do as a side effect is we cluster together dependency graphs that have similar meanings. So it will not surprise you that saying drug is an antibody directed against gene is semantically used similarly as D is an antibody targeting gene. Drug is an antibody targeting gene. These are semantically very similar, and they cluster together in this biclustering. So we get a bunch of equivalent sentences, if you will, that are then clustered together into these biclusters with the genes and drugs that have that relationship. So it allows us, you can see everybody in my lab enjoys this graphic. It allows us to draw this, I think, spectacularly exciting graphic, which shows what I call the universe of gene-drug relationship types. When you do cuts on this hierarchical graph of all of the gene-drug relationships, and I'm not talking about the hierarchical graph, but we can build a hierarchical graph in a pretty straightforward way, we see something like 25 modal interactions between genes and drugs as expressed in text. And I can tell you for every gene-drug pair which cluster they're in. And they're all color-coded, and you can see in some cases we've broken them up into two very subtly different sub-clusters. But let me give you a feeling for what they look like. To the first approximation, I would argue that this is every possible gene-drug relationship that is seen in text. So, gene synthesizes drug. That's for drugs that are like proteins or biologics or natural small molecules. Gene activates drug. Gene enzymatically modifies drug. I'm not going to read them all to you, but these are the kind of labels we can put on these colorful clusters that we've identified. And it's extremely satisfying for me because I know that everything I ever want to say about a gene-drug relationship is probably on this page. Now, what can we use it for? That's all great, but so we took Drug Bank, and Drug Bank has a bunch of drugs that have their targets, and we plotted in blue all the target relations in Drug Bank. So you can see there's a height over here, this fuzzy, bushy blue. That means there's a lot of targets in Drug Bank that are in these clusters, and then there's a little subset over here. Those are the clusters that allow us to predict new targets by looking for gene-drug pairs that are falling within those same clusters, but that are not currently in Drug Bank as a drug-target relationship. And when we look at we've made several of these predictions, hundreds of them, and when we go back and manually annotate them, there's a very high true positive hit rate. Similarly, for PharmGKB, my database, we look at drugs that have been annotated with genes where the genes modulate the genetic response to the drug. And again, we see a distribution of some clusters, partially overlapping Drug Bank, not surprising, but some of them not overlapping Drug Bank, which are the sweet spot for finding unannotated new gene-drug relationships that might be of pharmacogenetic significance. That's why I care about it, because I can give these to my curators and say, could you please look into these? Because after a lot of work, we have really good evidence that these guys are going to be important for PharmGKB. That's the end of the second story, and I think we're okay on time. Let's go down to gene expression. We're very interested in the effects of drugs on gene expression. As you know, you take a drug, if you're swallowing the drug, all of it goes to the liver first and then throughout the body because of the way the gut circulation works. We were interested in what are the genes that are like the first responder genes in the liver. Is there a subset of genes whose expression is controlled substantially only by the chemical makeup of a drug or a small molecule? And so how would you answer that question? It's to look for gene expression changes that are correlated just with the chemical features of a small molecule. And that's what we did, and this is work that came out last year, Guy Fernald, as part of his PhD dissertation. So here's a model. We have drugs that have little colorful glyphs on them. They bind to a number of receptors, either on the surface of the liver cell, or perhaps they translocate inside the liver cell. Then there's a lot of biology. That's the dashed lines stand for biology happens, not otherwise specified. But it leads to expression changes that lead some genes to go up, some genes to go down, as part of the gene expression program for xenobiotic. Xenobiotics just means foreign substance, sorry. Leads to the xenobiotic response of the liver, which is in charge of metabolizing foreign substances and basically getting rid of them. So we used the old Iconics company database, which is now in the public domain, the drug matrix, which measures rat liver expression. It's an old data set, but it's remarkably good quality in our opinion. It measures rat liver expression in response to 170 drugs. And we found 3,400 genes for which we had enough data, that is to say at least 20 drugs were upregulated 1.5-fold or more in response to all 3,400 of those drugs. So that gave us at least a training set to perhaps be able to predict the ups and downs, or in this case, the ups of these drugs. And so there's the kind of a rendition of that data. So we now we dove into the field of chemoinformatics, which some of you may be working in, and we represented our drugs with extended connectivity fingerprints, which basically take a small molecule shown here on left, and with increasing radius in terms of number of bonds, find the fragments centered on an atom. So here we start with the oxygen, and we have the diameter 1, 0, 1, 2, 3 fragments that are contained within this small molecule. Then they're hashed, and you turn it into a huge bit vector, 19,000 features to cover all of drug bank. So that's huge, but you have this bit vector. So the first problem that Guy had, of course, is that is way too big for machine learning. You're just going to overfit. So he did a PCA, very straightforward feature reduction technique, which many of you have, I'm sure, used. Did a PCA on this 19,000 features and just picked the first 20 components of the PCA. And so every drug now is represented by 20 coefficients that are its best fit to these 20 principal components that describe its chemical structure. So it's still a chemical structure description, but we've boiled it down to 20 numbers to kind of avoid some of the issues of overfitting. So then what do we do? Well, we have 170 drugs. That's what the rows are here, each characterized by 20 numbers. And we have 3,380 genes, either up or down in response to that drug. And I remember I told you that we made sure that there were at least 20 genes that made 20 drugs that made each of these genes go up in each data set. So we did machine learning. We did a simple LASSO regression to see which genes, if any, we could predict solely based on the chemical features of the small molecules that induce them. So first I should highlight the one on the right. When you randomize the data and look for and try to build classifiers, that you get, here you have 3,800 classifiers, each built 10 or 20 times. That's why you get the gray variation. And you can see the nominal significance of 0.05, and that's kind of our random background. When we look at the real data, we focused on a small set of genes in the upper right that are consistently above the significance cutoff for multiple runs of 10 or 20, I can't remember, 10 or 20 runs of the regression, rebuilding the regression and retesting. So in the end, we concluded that of these 3,300 genes, 87 of them, we think we actually could predict whether expression is up based only on the chemical structure, which is kind of maybe what you might expect. I didn't know what to expect. Here are the top 10 genes in rat liver whose increased expression we can predict from chemical features alone, and you can see the area under the curve on the far right. So by far our best is cytochrome P450-1A2, where with an area under the curve of 86.86. There's a lot of other famous genes here involved in the pharmacokinetics of drugs. And many of them you would not be that surprised are responding to the chemical structure because they have a certain specificity for certain drugs, and they are probably responding to the specificity, to the drug features that are, we would love to know the biology that connects them, those dotted lines, and that's for the future. One that we were very intrigued by the way, is number 8 here, or 10 or whatever. The VLDL receptor seems to, at least in rats, be upregulated in response to certain drugs. And so we're pursuing that because that could be a drug target. It could be a way to screen for new drug leads against cholesterol. Just to show you how we use this, for that best performing gene, CYP1A2, there were 3 drugs that were not in our training set that had recently been reported as inducers of CYP1A2. That is, they increased the expression. So we ran them through our classifier. What you can see is the blues are our positives in our training, the oranges are negatives, and you can see the scores that were achieved by these 3 recently reported inducers of CYP1A2. So that was a good confidence builder for us, because it meant that even in prospective application, and we had a couple of other prospective applications, this seems to be working. So this was a fun project for us, because so many of us are using gene expression to learn about other things, but in this case we were actually trying to predict gene expression. We cannot definitely do it generally, but for those 80 or so genes, it seems like the identity of the chemical ligand itself is in large part sufficient to recognize an induction. And of course, you can look at these PCA components and figure out which fragments are contributing the most. So what you see here are the fragments associated with positive induction. So these are positive features for the induction of CYP1A2, and then the orange ones are things that go against CYP2 induction. So you can get a little bit of chemical insight into the features of the molecules that are causing these expression responses. And of course, there's more to do there as well. So that ends the third, or the penultimate. That's the penultimate. So now I'm on my final story. That was at the cellular level. And again, all of this in some way winds up back in PharmGKB, either informing our design decisions, or actually we just put the databases up on PharmGKB, or we give advice to our curators about where to focus their attention. In this last project, I want to talk about some recent work. Just came out in one of the society's journals, PLOS Comp Bio, Grace Tang, her PhD thesis. The knowledge-based identification of fragments likely to bind protein pockets. The headline here for you is that we don't use physics at all. I love physics, I believe in physics, but I'm really a little bit more of a data person, and there's now enough 3D structures in the protein data bank and other places that I think we can take machine learning methods towards predicting drug binding in pockets of proteins. So can we use the full database, the full protein data bank database, to predict at least fragments that will bind into a pocket? And then can we use those fragments to either design drugs de novo or screen libraries that have these fragments as high likelihood candidates for being inhibitors or binders to the pocket? So we have this program called FragFeature. It's based on the feature tool, which our lab's been working on forever, and I'll describe that for those of you who have never heard of it. But the idea is we have a query protein with a pocket. The pocket is shown as these colorful balloons. I'll tell you what those are in a second. Each of those balloons, though, represents an environment or a microenvironment of a protein that has been observed close to a set of fragments. So what you see here is the knowledge base, which associates different microenvironments with the fragments that they like to be near. Now, I want to stress that even though this is a serine part of the knowledge base, it's not a single serine. It's not serine likes to be near these fragments. It's an entire environment around serine, a particular environment that I'll tell you about in a second, likes to be near these kinds of fragments. Whereas another kind of serine environment likes to be around these oxygens. We take this knowledge base, apply it to this pocket, and look for combinations of these colorful balloons or microenvironments that give us a strong statistical signal for a fragment. So it's really a ridiculously simple idea. Just use statistics to find the association of protein neighborhoods with fragment binding preferences. So how do we do it? How did Grace do it? Well, the first thing is we take all co-crystals of 3D structures that have a small molecule ligand bound. So here you see two ligands bound into two cartoon pockets. Notice this ligand is an adenine, has an adenine in it, and this serine, the square serine, is very close to all of that adenine. Whereas this serine, the circle serine, is actually close to a slightly different set of fragments. That's what I was talking about where this is not just about serine, it's about the environment of the serine. So we look at the corresponding feature microenvironments and accumulate the statistics of their proximity to fragments. Now I need to tell you about feature. There's a 3D protein structure into which we pick a central point and we define 6 or 7 concentric shells. And in each shell, we count the occurrence of chemical and physical features, like the types of atoms, the types of charge, the types of amino acids, the types of secondary structures, so that each of these spherical environments becomes a vector of about 400 numbers. And this is actually probably the only good idea here, is that how do you take a complex three-dimensional environment and linearize it into a vector that you can then manipulate? And the answer is each shell provides 60 to 80 features. We have 6 shells. That's 360 to 480 linear numbers, most of them either integers or real numbers, that we can then use to describe the three-dimensional environment that you saw circled by this little blue cloud. So that's called a feature vector. And each of those balloons that I've been calling them is really a vector of 400 numbers summarizing that space. And we've used this literally for 10 or 15 years and have shown its ability to distinguish active sites and that it has real information content. And that gave us the courage to do these experiments in drug design. So literally for every co-crystal in the PDB, we look at all of the ASP feature vectors that are close, and we write down all the fragments that they're next to. And so we have this huge knowledge base of hundreds of thousands or millions of feature vectors associated with the fragments that they like to be near. Okay, hopefully I haven't beaten a dead dog. So now we can actually use it to do something exciting. We take a new protein, we identify its pocket or pockets, and we calculate all the feature vectors shown here as the colorful balloons. For each feature vector, we look up its 5 nearest neighbors. This is not fancy stuff. It might become fancy, but right now it's nearest neighbors. K equals 5 nearest neighbors, and we get the list of fragments that they are close to. Then we do a little hypergeometric p-value because you have to kind of account for, this is like balls and urns, and you have to account for the balls that are in there frequently so you don't always predict common fragments. And there's a bunch of statistics that I won't talk about because it's the last talk of the conference, but you have to do some careful thinking there. We do that for one ball, but notice that this pink ball, this pink balloon, is near a blue and a yellow one. So we also do it for the blue and the yellow. And so they all have their votes about the kind of fragments that they want to be near. And that tends to give us a very strong signal for a few fragments that we would predict are the fragments that this little piece of the pocket wants to be near. Let me stress a couple of things. That's a big pocket with, as shown here, 9 different feature vectors. So we're just predicting that at this end of the pocket, these kinds of molecules have a high likelihood of binding based on knowledge-based comparison with other structures. So, of course, we validated this on a bunch of famous molecules, adenine, ADP, NAD, vitamin B receptor is at the bottom there. That was our worst performance. In general, the recall and the precision, I'm not going to go over it in detail here, but in general, they were pretty good, and they gave us confidence that we could go forward with application. So let me show you an example. This is exotoxin A. It's a bacterial toxin protein from Pseudomonas, and it is an ADP ribosyl transferase that interferes with protein synthesis. And it has this lovely pocket that has what? About, I don't know, 9 or 10 feature vectors in this pocket. When we run that little pipeline that I told you, we look at the preference for each of those, we get a very strong signal for I think that's a benzamide fragment. One of the nice things we can do, and this is very useful for our 3D structure collaborators, is they say, well, why do you like that? We can find those nearest neighbors that were used and show them. And so here we can say, well, here's 6 protein structures that have equivalent environments. Here's the blue balloon in our structure, but there's a similar blue balloon, a near neighbor in this other PDB structure. There's a couple of pink ones, there's a red one, but they're all near these small molecules that contain this fragment. So it's very good for explanation and for justification of the prediction, which for anybody who's ever worked with a biologist is critical. And in fact, we can write down all the ligands bound by their nearest neighbors, and what do you know? Almost every one of them has this small chemical group. And in fact, in one case, that molecule, which was bound to a near neighbor, it had actually been tested against this protein in Chembl, and we know that it had 140 nanomolar binding. So that was a good confidence builder. One other example, atypical protein kinase C. We and everybody else in the world are very interested in protein kinases and kinase inhibitors. I love this example because it's a big pocket, and one half of the pocket likes this fragment, kind of a 6-membered ring with a nitrogen. I'm sure there's a chemical name, and I apologize for not knowing the chemical name. And the other end of the pocket likes just a good old-fashioned 6-membered ring, but with very high score. When we look at 3D structures, other 3D structures of the same protein that were not used, obviously, in the training, we are very lucky because there was a bound ligand to this same protein that contained the predicted fragment on the left, and there was a different bound ligand that had the predicted fragment on the right. And I want to point out that you can see that this purple fragment is at the far and back right, coordinated with the orange, green, and reddish environments or balloons or feature vectors, whereas this one is up in the front and on the left. And so the immediate thing that we're in the middle of doing is, are there small molecules that have the red thing and the purple thing connected by an appropriately length linker? We would predict very tight binding if such a molecule exists. Or if one of you medicinal chemists in the crowd wants to synthesize that for us. So a very exciting example, and I won't say anything more about it. There's other things that make it exciting, which I'll skip over. So those are the 4 stories that I wanted to tell, and I'm glad I left some time for questions and for the end of the conference. But I hope I've convinced you that, first of all, PharmGKB is a good resource. You would have to take my word for it because we hardly spent any time on it. But what it's great for is by generating an excuse for us to do these kinds of studies to support our understanding of the molecular basis of drug action. I'm very excited about informatics in the context of many scales, inferring and triaging side effects from population databases. That gives us hypotheses that we can bring to the molecular and cellular folks. Can you see a molecular reason for this side effect? Can you see a molecular reason for a cellular reason for this side effect? Are we seeing expression changes in gene pathways that explain this side effect? This is one of the things. And I should say, when I say side effect, I really mean any effect, because today's side effect is tomorrow's repurposed drug. So you should always use side effect with a grain of salt. Can we characterize gene-drug interactions from text, just so we know what's possible, and so we don't miss things that are contained in text but not curated by our database? Can we predict expression from chemical structure? And we've shown we have. And how can we use 3D structure to predict fragments in the context of either new drugs or screening existing small molecules? So the combination of these data sources I'm very bullish on as powerful data sources for understanding pharmacogenomics, for determining the drugability of new targets, for discovery of new drugs, and for repurposing of old drugs. So with that, I will stop. I will thank you for your attention, especially here late in the conference, and I want to thank the people who work on PharmGKB and the people whose specific work I discussed today and funding sources. Thank you very much.
Are there questions? Please step up to the microphones.
Dr. Searles.
Very nice, Russ. I'm curious what reaction you get, the fourth part of your talk, when you speak to medicinal chemists, and in particular, have you compared those results to standard pharmacophores that are already well known, and have you thought about exploiting that with the current approaches to fragment-based screening?
Yes, so all of this is on our radar. I have nothing to report now, but we're talking to colleagues in industry who have the ability to do this screening and who do kind of physics-based pharmacophores and pattern-based pharmacophores. And we think, we don't think that this is the be and all end all, of course, but we think that it's extremely complementary to those approaches because it's based entirely on the observed data. One of the great things about drug companies is they have, as you know very well, their own database of 3D structures enriched for co-crystals, which would make our algorithm work even better. So I'm very interested in those kind of collaborations and for comparing it. So far, what we're focusing on is we've made some predictions that are being tested in the lab because we thought that would also be very compelling. So for some of these exotoxins and stuff, we have very strong predictions that are somewhat non-obvious, and we're testing those with plasmon resonance experiments and binding experiments. Not we, but paid collaborators.
Thanks for a clear talk. What do you think is necessary to extend the side effects mining work to third world countries?
So when you say third world countries, you mean the third world countries because?
Well, let's say tropical diseases, like
yes.
Ebola.
Right.
Right. So there's a epidemic right now for Ebola, and people are just refusing to believe in reporting them.
Right.
Right. So what do you think is necessary for that?
So the first thing that's necessary we have, which is the almost universal penetration of cell phones. And as I'm sure you know, in the infectious disease world and in the reporting world, the availability of Twitter and tweeting and social media has given access to, it's great to look at, Boston is having a flu outbreak, big deal. But to know what's happening in these countries based on the self-reports, I think that's a great starting point, but what has to happen then is an infrastructure has to be created for maintaining quality of this. In our crowdsourcing, we spent a lot of time on these QC queries because we knew that it would be garbage in, garbage out, and we had to throw out some non-trivial, I think it was 9 or 10% of the data because they didn't pass the test. So I'm kind of optimistic that crowdsourcing in the developing world for drug trials and for disease outbreaks might be certainly an hypothesis generator. Now then over a beer, you have to argue about whether crowdsourcing can prove things or just suggest hypotheses that require more old-fashioned epidemiology.
All right, thank you.
Thanks.
Hello, thank you very much for this inspirational talk. Seeing antibacterial medicine at the lower end of your side effects list made me think that maybe if we look at side effects on a population scale, for example, misuse of antibiotics in a country can cause antibiotic resistance and things like this. Maybe your database could be widened to a larger scale to see these.
It's a great point, and I should stress, because you make the point, all of these are kind of side effects that are observed at the individual level. So if you count total resistance to all known antibiotics as a side effect at the social level, that should be very high. And as you know, there are certain infections that are worrisome because we are down to one effective antibiotic or one or two effective antibiotics. And for a variety of financial and political and social reasons, development of antibiotics has not been, as you probably know, a high priority at many pharmaceutical companies. And so we're looking at a period where we could have a problem. I think this is exciting for academics because it gives us an opportunity maybe to make some contributions. All of the things we're checking in the lab are bacterial proteins that we think might be anti-infectives. And the reason we chose that is because we know that there's this need, and these were non-trivial predictions of novel mechanisms. But you're absolutely right that the work that we're doing is for the individual side effects, and there's a parallel analysis you could be on the public health implications of drug use. We are doing a little bit of this work for pharmacogenomics. I've spent so much time on pharmacogenomics that I'm interested in getting it implemented before I'm dead. And there's a lot of people saying, well, where are the clinical trials that show that it's efficacious? And I'm pretty sure I will be dead by the time those are done. So we're looking at ways to build models of the impact of pharmacogenomics on patients and at least show with confidence bounds the likelihood that these interventions would help more than hurt. And in my heart, I believe they will help more than hurt, but it would be better to have something more than my heart. And so in a totally unexpected turn, I have students talking to health economists about how to do these kinds of population level assessments.
Thank you.
Thanks.
Thank you very much for this exciting lecture. And my question is, so you have investigated the interaction with gene and drugs, and you looked at relationship with gene, drugs, and age, and sex, and so on. But there is also food and diet. And
Yes.
Have you thought about
Food and...
because the relationship and the interaction should be a bit more tricky.
So thank you for that question. First of all, when you talk to a patient, I'm a doctor, I don't know if I mentioned that. I actually talk to patients every now and then, which is scary for them, but they don't know that I mostly do this. When you talk about the things that cause variability in drug response, yes, genetics is one of the factors, and it's an important factor, but what's in the patient's stomach is an incredibly, probably dominant factor because they can affect absorption. Whether the patient actually fills the prescription and takes the medication is pretty darn important, and that actually would be another thing to work on. And then the microbiome. So if you would allow me, I would add food and the interaction of food with the microbiome as huge areas that will probably explain, in my opinion, much of the unexplained variation in drug response that we're having trouble explaining from genetics. Genetics does pretty well in some cases of explaining variability, better than diseases in many cases, but it's still not enough for a clinician. And so I think we're going to have to work this in. And so I'm very interested in microbiome issues, the impact of food. And so we have no results in that area. I have reviewed some papers, maybe by some of you recently, very exciting in this area. So you might be seeing them soon in a journal near you.